All right, so let's get started. Thank you very much and welcome to the EPAASM monthly meeting. My name is Vincent Tam. I'm the branch president, and thank you very much for spending your time here with us. I know it's a hassle and this previous game, but thank you for being here, and I'm sure you will enjoy the hour. This is a very special meeting. This is our James Poupard Memorial Lecture, and thank you, Barbara and James families for being here. And before I start, just to say that we're a branch of ASM, and ASM is a community of microbiologists. And so show your support. We join or renew or join the national ASM membership. And this is the way to nurture this and grow this community. And ASM also has a Center for History of Microbiology and ASM Archive. That was in big part due to Jim's contributions. And so you can look at all the history of microbiology on the website. Of course, the National ASM Microbe Meeting is in June 2024 in Atlanta, Georgia this year. You can submit your abstract in November 15th starting. And each year we have 3 travel awards, Peggy Carter Awards to give out within the branch that will cover part of the travel to ASM microbes. And we'll open that applications and nomination process soon. And we wouldn't be here without the support of our members. We are one of the oldest branch and certainly the most active branch around. We've established back in 1920, so more than 100 years. And we're here because of support of our members. We have a very active student chapter. We have an education committee and an executive committee. So as you're here, so you know that we have a monthly meeting programs. And every year we also have Philadelphia Infection and Immunity Forum, as well as the clinical symposium. And we have a lot of scientific outreach and education programs. And just to highlight some of those things, this is our upcoming events for the monthly programs. So we have our Infection and Immunity Forum on December 1st and the clinical symposium December 8th. And in January, we have our industry speaker. And then in February, we have our own Christoph Theis from Penn, Camille Lesser from MGH in March, and then our ASM distinguished lecturer, Joanne Engel in April. And so PIF is coming up. It's on December 1st, and abstract, you can submit it. The deadline is this Friday, so get your abstracts submitted. And we have a great lineup of keynote speaker, as well as we will have trainees, graduate students, and postdocs giving talks and posters. And our clinical symposium is also coming up. This is our 52nd clinical symposium. Again, Jim was also a big part of chairing a lot of those symposium. And again, this year, the theme is back to the future, new technology. And so we have a lot of NGS as well as AI utilization in clinical microbiology. And again, registration is open, so go to our website and you can register. And I also want to highlight some of the great stuff that our education committee is doing. Linda and Pina recently partnered with iPraxis, and they went to Lane Lock Elementary and do a Gutsy Microbes, where they talk about diet and microbes to elementary school students. And they also have an activity for making yogurt. And they did it on October 11th, and they're planning to do 12 classes per year. So these are just some of the great stuff that we're doing in all the different sections. And so please support the branch. Go to our website. And for faculty and postdocs, it's only $20 a year that pay for everything, including the food and drinks outside. For students, actually, it's free. So you can, I'm not advocating that here because I'm being recorded, but you can join the branch without joining the national ASM membership. So please do that. It's open now for the whole year of 2024. So I would love to introduce Linda Miller. She was our branch president from 93 to 95. She was a student, a mentee, and a colleague and friend of Jim. She is very dedicated to the branch. Since she's been the chair of the clinical symposium, she's been part of the education committee as well as the archive. And without further ado, Linda Miller.
Thank you, Vincent. I just want to welcome all of you here and all of you on Zoom to the second annual James A. Poupard Memorial Lecture. I want to give a special welcome to the Poupard family, to Barbara, and to Jim and Barbara's children, James, Nanette, and Jackie. And a grandchild is with us tonight too, Jen. So thanks to all of you and the whole Poupard family. For those of you who did not know Jim, Jim was a clinical microbiologist, a historian of science, an associate professor, a mentor, a husband, a father, a friend, and a lifelong Philadelphian. So I am not going to go into all the places over 34 years that Jim was in some kind of education in Philadelphia. His bachelor's degree was from Temple University. His master's in clinical microbiology, first in the class under Dr. Eileen Randall, was at Thomas Jefferson University, and his Ph.D. was from the University of Pennsylvania. So Jim's career can be described as interdisciplinary, eclectic, and he was certainly curious. His early career, he had many different positions in a variety of laboratories over the years while he was still going to school. Those included Penn and Wistar. And I can't go into all the details, but we are putting a lot of this information on our website. But it was really a very interesting and eclectic career early on. And then he focused onto clinical microbiology. After his PhD, after his master's and PhD, he came to the Bryn Mawr Hospital and was the director of clinical microbiology there. He then moved to the Medical College of Pennsylvania, where he directed the clinical microbiology laboratory. He was an assistant professor, the medical school, and the graduate school, and he directed the graduate program in clinical microbiology. He then moved on into industry to the anti-infectives research therapy area, and led a number of different groups in that area under SmithKline Beecham, which then became GSK. And after leaving there, he created a consulting group that consulted with biotech companies. So Jim had over 100 different scientific publications. He was an recognized expert in clinical microbiology and also in a number of areas. He was also a fellow of the American Academy of Microbiology. And you can see a variety of his research areas where he had published papers. But I also am going to just highlight that one of the areas that he did some work in was in novel approaches to answer epidemiological questions. And this is relevant to similar, in some ways, to what our speaker is going to be talking about tonight. His focus was on strep pneumoniae and also antimicrobial resistance in these studies. As Vincent said, Jim was a member of our branch. He was a member of our branch for over 50 years at ASM National and our branch. He was our 36th branch president, served on many different committees. And I did note that I have a different number, being the archives committee now. Like, okay, one of our numbers is wrong. I will grant you that it's probably my number, which is embarrassing. Jim was the branch archivist for many years, and he contributed much of the history content on the branch website. And for ASM National, he served twice as the chair of the Center for the History of Microbiology and ASM Archives. And we have a lot of information about that, again, that we're going to end up putting some of this on our website because it's really, really interesting. In 2010, Jim published a book, A History of Microbiology in Philadelphia, 1880 to 2010, which is a really great compilation of microbiology in our city and what an important center Philadelphia was in this area. It also includes a detailed history of our branch. And this book is really something that we treasure in our group, in our branch. So Jim had an infectious enthusiasm, not only for microbiology, but for science in general, for quantum physics, philosophy, and history. His passion for life endeared him to a very devoted following of students, friends, and colleagues. And Jim believed deeply in the value of this branch as an incredible opportunity for young people to get to gather and meet and share ideas with so many of the leaders of our amazing microbiology community. So it's a real special opportunity. Jim introduced me and many medical technology students, medical laboratory scientists, graduate students to the branch. He would organize carpools to drive us down to the meeting and to get us here, so that, which were at one point, and for most of those years, was at Jefferson Alumni Hall. And I think that some of you here will remember, and some of you on the Zoom, that after every December meeting, there was a pancake and scrapple dinner at the Poupard family house, which was graciously hosted by Barbara. So we gather here tonight as a memorial to Jim Poupard, and we're grateful and honored to have Dr. Nathan Grubaugh as our memorial lecturer. I am sure Jim would have been excited about tonight's talk. We're very much looking forward to learning from you tonight. Thank you.
Great. As Vincent switches, I'm going to introduce our speaker this evening. So it is my absolute pleasure to introduce tonight's speaker, Dr. Nate Grubaugh. Dr. Grubaugh received his bachelor's in biomedical sciences from Western Michigan University, his master's in biotechnology from Johns Hopkins, and a Ph.D. in microbiology from the Colorado State University. He completed a postdoc in virus genomics at Scripps before joining Yale School of Public Health in 2018, where he is currently associate professor of epidemiology. I was introduced to Nate during the pandemic, where he was leading a number of projects to monitor SARS-CoV-2 genomics, including tracking viral spread and transmissibility. We actually submitted data together that resulted in a paper that was written and submitted in 2 weeks, and the fastest paper probably either of us have ever put together, that included 7 different institutions. So that was quite a treat during that time. But I think the most interesting work that he did, at least in my opinion, is that he tracked genomic epidemiology of SARS-CoV-2 for the NBA and the NFL, which is a really cool thing to get involved in during a pandemic. He's been widely recognized for these efforts, including winning the On the Front Line Award for the Association of Public Health Laboratories. But more in line with tonight's lecture, and I think more in his traditional area of expertise, Dr. Grubaugh has applied his skills to studying vector-borne viruses, including dengue and West Nile. He is the recipient of an NIH Director's New Innovator Award and an NIH NIAID grant to study dengue virus genetic diversity. And recently it was announced that he's the recipient of the ASM Award for Early Career Applied and Biotechnological Research. So without further ado, please welcome Dr. Grubaugh.
Well, thank you for that very kind introduction, and thank you all for being here. It's quite an honor to be giving this lecture. I didn't know Jim personally, but I can say that clinical microbiologists are some of my favorite people, and especially during the pandemic, I've learned that they, as an epidemiologist, are our connection to the hospital to be able to answer really important questions in public health. And well, here I'm going to talk not so much on the clinical side of things, but certainly all of our work with COVID involved a very close relationship with those folks. I think this is now officially the largest mosquito I've ever seen. Certainly the largest screen I've ever presented this on. I'm not here to talk about COVID, but a little bit of a backdrop in all of this, because it obviously is important in a lot of the ways that we think about questions and technology and collaborations and such now. So here, hold on. All right, so in general, my group focuses on how can we use genomics to help answer questions about disease ecology and epidemiology. And I hope what you'll see from this, we had to convince a lot of people early in the pandemic why sequencing SARS-CoV-2 was important. Why was genetic diversity? It was kind of in some ways, still people think about it as like stamp collecting. Like, okay, cool, you're doing this, you're getting some sequences, but what does it really mean? What we're trying to do is use sequencing and routine sequencing and phylogenetics and the approaches that I'm going to talk about today to reveal important aspects about how viruses in this case can persist in our environment, how they can cause disease, and can we find little shortcuts or some aspects of their life cycle that we can exploit for control. So here's just an overview of some of the things you can use with genomics. To start with on the, here, can you see this? Oh, great, this works. Here, detection. Thinking about the pandemic again, it was metagenomic sequencing of some of the early cases of pneumonia that identified that it was a novel coronavirus. The sequencing here went from like something that was syndromic, not sure exactly what it is, to actually being able to figure out, okay, this is a new coronavirus. We know something about coronaviruses. Can we use that information? To a few sequences early on as something is emerging can help us understand, is this a new event? Is this a new introduction in the human population? What is it related to? Based on those things that are related to, what does that tell us about the epidemiology? And as we get further in, if we do dense sequencing in certain environments, we can use this to understand transmission chains. Kyle talked about the NBA and the NFL. We sequence every single case in these leagues to help to do transmission chain tracking, so we can understand if their protocols are working correctly. And actually, we're able to apply that to a broader level to understand in our communities, not just in these sports environments, are masking and things like that actually working. And then finally, on a larger scale, we can use genomics to help track the emergence and spread of pathogens as they go beyond international borders. We've spent a lot of time setting up these really massive sequencing campaigns for SARS-CoV-2. There's been something like 4,000 different labs that have generated SARS-CoV-2 sequencing data during the pandemic. I mean, close to like 20 million sequences now. So much infrastructure that has been put in. And now, let's not turn those pipelines off. Let's point them into other directions where we can get this information to actually be useful. And so this is what I'm doing now is using these SARS-CoV-2 sequencing pipelines to get data from viruses that need more love. So this talk, I'm going to go through 3 vignettes. If we don't have as much time for the bottom one, we can kind of skip through it fast. It's fine. That really highlight the things that I really care about. I love complex ecologies. I love working in systems where we don't know all the answers. And things that are also really important for public health. So starting with Powassan virus, this is a flavivirus like West Nile and dengue that is transmitted between ticks and mammals. This story is going to take place in the Northeast. and then also then a mosquito-borne virus called Eastern equine encephalitis virus. And then finally, another mosquito-borne virus called dengue, where we're going to focus mainly on the Caribbean. So to start, this work on Powassan virus was led by an associate research scientist, Chantal Vogels, in the group that is now moving on to her own faculty position. We published this paper this year. We started this project before the pandemic and then started to pick up on it again recently. A bunch of different collaborators. If you were to look through the actual affiliations here, you would see a lot of public health departments that we work on for this project. So Powassan, who has never heard of Powassan virus before? To be honest. I think this is one of the most important viruses that you've never heard of. And I'm not trying to be like, scary or whatever. This is transmitted by the same ticks that transmit Borrelia that causes Lyme disease, Babesia, Anaplasma. It's called the deer tick or the black-legged tick. These ticks live for several years. The eggs are hatched in the spring, and then you have larvae, and then a year later you have nymphs, and then finally adults. And to go through each stage, they have to take a blood meal. And that is important because that's where transmission occurs. I'm going to talk about, so there's two lineages of Powassan virus, and this is important because they have different ecologies. Lineage 1 that I'm not necessarily going to focus on here, this is primarily maintained in a lot of different small mammals, woodchucks, squirrels, and the tick is Ixodes cookii. And this is important because they don't really feed on people. So there's not a whole lot of human cases of this lineage of virus. On the other hand, lineage 2, this is where I'm talking about with this transmission cycle over here, where it's maintained by Ixodes scapularis, in which will feed on people. And this is where we get, again, Lyme disease from. Also, lineage 2 of this virus has two main foci, one in the Northeast, and from what I understand, also here in Pennsylvania, and another one in the Midwest. And these are genetically distinct, as you can see from here and here. And so the focus of what I'm going to talk about here is what is happening in the Northeast with this lineage 2. The cases of this virus are increasing, in part because there's increased detection for it, increase in awareness, but also because there's increased exposures. These ticks are actually rapidly expanding. This is a very climate-sensitive system. And the other aspect of this is that the cases that we do find are quite severe. This causes neurological disease, oftentimes can lead to death. But we know almost nothing about its ecology. We don't really know how this emerged in the Northeast, when it emerged, how fast does it spread. We don't really know how it's maintained. So to exploit aspects of its life cycle for control, we don't yet have the information. So that's what we set out to do with this project. So someone who does a lot with genomics, we started off this project, we go into the public repositories and say, okay, asking what data is available here. And when we started this, there were 38 complete genomes of this virus, which wasn't a whole lot to do much with. And so over the years, we built up collaborations in many different areas to get additional samples, mainly from tick surveillance programs. So none of these are coming from human cases, but they're coming from actual positive ticks. And we've been able to build that up to over 400 now that are available, and this project is still ongoing. So what can we do with these data? So this shows a time-resolved phylogeographic tree, meaning that each one of these circles here, which we call tips, this is a sequenced virus, and it is placed on its collection date along this axis here, which is time. The lines represent how things are genetically related, and where they come together, like here, this is the estimate both in color of where we think this ancestor occurred And when do we think it occurred by looking down? And one of the things that the first thing we can get from this analysis is that of all these viruses in New England, their most common recent ancestor existed in about the 1950s. So that's when we actually think that it emerged in the Northeast. It's not that old of a disease. To speculate a bit about the ecology here, so we think that maybe Borrelia that causes Lyme disease came in some years before that, and before that would have been the emergence of these Ixodes scapularis ticks into the Northeast. And what these ticks have followed is the reforestation in New England, which allowed for the white-tailed deer, which are important hosts for the adult ticks. And then eventually the ticks came. And here it just shows the deer population in Connecticut and New York and how they're increasing around this time in which they emerged. I'm from Michigan, and I didn't know anything about like the reforestation that happened in New England many years ago. And so I had a lot of fun learning about this. Does anyone know why it was deforested and why it was reforested? If you have an answer, just shout it out.
Building log cabins.
Log cabins, all right. Farming. But why was it reforested then?
Industrial revolution.
Farming moved to the Midwest. All of New England was primary farming land for a while. When I go for a hike in Connecticut and I walk through forests, which is funny to see are all these old stone walls that used to outline fields. So farming was one of the major reasons why that when that went to the Midwest, then there wasn't as much of a need to have farming and the trees grew back. But another thing that happened too, which it was interesting to learn about, we were talking about a hockey team, the Hartford Whalers that used to exist. Whaling industry was very big in New England. And with that was building these wooden ships. And when the whaling industry thankfully collapsed around the late 1800s, and then the forest started to regrow somewhere after that, then we had an introduction of deer and ticks and virus. So there's complex interactions of the way that we shape our landscapes and how that impacts the diseases that are present. Anyways, I'm not that much of an ecologist. I just find this a really interesting story. There's also a whole bunch of relationships around acorns and Powassan that I won't get into, but I'll let you think about that. All right, so, sorry. If we take this tree and we have location and time that we can estimate, we can put that onto a map. And so that's what we're doing here. And each one of these circles is where we have sequences from, and the lines connect them in space and time. And the main thing here is that what we see is this spread from south to north, right? And that's where, and it follows the pattern of the deer and then the ticks moving up. Makes sense. What was really interesting here is that we see a lot of slow local spread intermixed with these sporadic long distance introductions. So mostly it's not moving very far at all. And then all of a sudden gets shot way up in different locations. And what we think is that when we have infected larvae or nymphs, those stages of the ticks that then are accidentally feed on a bird, the birds can then take them and they can travel long distances. This doesn't happen that often, but it happens enough that helps to move its spread along. And with that, we have, again, we estimate in parts of New York and southern Connecticut, this introduction in the 1950s and making its way all the way up into Maine by about the 1990s. And this continues to make its way north. And so with increased surveillance for this, we'll probably start to see more human cases as it keeps creeping up. To put this in perspective, this shows what is called the dispersal velocity. So how fast are these viruses moving within infected ticks or animals over time? And this is just to show that here, Powassan virus that's maintained in ticks and mice, this is actually really slow. It's very similar to Lassa virus, which is a rodent-borne virus that is found in West Africa. where things like rivers can be a barrier that they can't get across for thousands of years. Once you start putting wings on the system, they move a lot faster. This makes sense. But just to put it into context, when West Nile emerged into New York in about late 1998, 1999, and moved across the continent, the dispersal velocity was 55 times faster than that of Powassan. This is very slow. This is just another way to look at these phylogeographic maps. And what I'm doing here is highlighting some sites in southern Connecticut. And each one of these dots is like the GPS location of an infected tick. And what we see here is strong spatial structure. So we find in these sites, there's very little genetic diversity. Where did my pointer go? Very little genetic diversity within a site, but a lot of genetic diversity between sites. And also you don't see a lot of lines connecting these things. So if this was West Nile and you're looking at this map, it would just be a giant mess because they move all over the place. So if we look at these two sites, Reading and Bridgeport, which are about 12 miles apart, back in the day when I actually used to run, this is about a half marathon. Maybe on a good day, I could do this in about 2 hours and 20 minutes, right? Deer can travel these distances, but yet the viruses between these locations have been separated for 33 years. So we know that deer are important for the adult ticks and the deer can move around, but yet the viruses aren't. So this tells us something about how the virus is being maintained, which allows us to develop some hypotheses. So what we are hypothesizing here is that once these ticks get to adults and they're infected and they're feeding on humans and deer, this is not important at all for the maintenance of the virus, or else we would see a lot more mixing. And an important aspect on that, so this is important for us of getting infected, but not for the perpetuation of the virus going forward. There's been experimental studies showing that adult female ticks that are infected can actually pass the virus to their offspring through the eggs. But again, if this were to occur in nature, we wouldn't see the structure that we do. So we think this is actually pretty rare that it happens. Instead, we think that the virus is maintained really on the backs of mice or other small mammals they might be feeding on, where you have infected nymphs that are at the same place and location as larvae that are a year younger, and they're feeding on the same host, and they're transmitting it back to a younger stage. These larvae get infected, they survive till the next year as nymphs, and then they transmit it back to larvae. So we think here's where all the transmission is occurring. So the ticks can only travel as far as their hosts, and these mice and other small mammals, a field can be a barrier. A road is a barrier. A river is a barrier. You have tree lines, properties, things like that. And this makes sense of why there's very strong spatial structure. It also suggests if we were able to find these hotspots of where there's Powassan transmission, if you were to control the virus there and suppress those tick populations, you're not going to get reintroductions of new virus. This is a little bit different than the Borrelia Lyme disease situation, where we think that the either this vertical transmission, this transovarial transmission may be more common, and so it can spread a little bit faster. But Powassan actually has might be a little bit easier to control. All right, so that's vignette number 1. All right, jarring change. So we're going to talk about Eastern equine encephalitis virus. Who hasn't heard of Eastern equine encephalitis virus? A couple. Or triple E, we'll just say to make it a little bit easier. We published this paper this year, led by an awesome postdoc, Verity Hill. We're mainly thinking about this story from 2019 that we also had to put on hold with the pandemic and then later got back into. So triple E is like a swamp virus. It's transmitted between mosquitoes and birds that live in marshlands, forested wetlands. The primary mosquito is called Culicida melanura, though other mosquitoes are involved. It likes a lot of passerine birds like robins and sparrows, and it's very different by every location that you go. Is anyone here a birder? Cool. Birders are unique people. I really enjoy working with birders when it comes to these sorts of projects because the bird ecology itself is really interesting. In every different location you go to with EEE, there's different birds that are involved. But anyways, then when an infected mosquito either bites a human or a horse, we're called dead end hosts, right? This is where we see the disease, and in the name equine, right? This is a really serious disease for horses. There is a vaccine for it. And it's also a very serious disease for humans, but we do not contribute to the onward progression of the virus. So 2019 was a particularly bad year for EEE. The most number of cases on record since I think like the 1960s were detected, with a lot of them happening in the New England region, but also in Michigan and some places along the eastern coast as well, and also in Florida. For me, in the fall of 2019, so right, this is like right before the pandemic was happening, but there was a lot of fear about this because these infected mosquitoes that feed at dusk, right around the time that kids have like baseball practice and soccer practice and such. And so there's a lot of concern of parents of even letting their kids outside at that time. If you want to divide a community, talk about spraying mosquitoes, right? That'll certainly get people on both sides of things. And it was like, they're very difficult conversations. Obviously, pales into comparison to the conversations we had during COVID, but there was, I think, quite a bit of interest in understanding more about the ecology of this, the disease. So in Connecticut, in 2019, we had 4 cases, and every single one of the patients died from this. This has a very high case fatality rate. This is obviously the tip of the iceberg. There's many more human infections that are happening that we never diagnose that are probably asymptomatic. But if you are symptomatic and have neurological complications due to EEE, we have no treatments for this besides basic supportive care. There are also several horse cases that year as well, and usually when you find a horse that is infected, that it has to be euthanized. So we wanted to know, again, basic information. How is this virus being maintained? And what drove this outbreak that happened in 2019? So what we did is we sequenced samples, as my lab does. EEE is a select agent, and even the RNA of this is a select agent. It's incredibly hard to work with if you do not have select agent status. If you detect it, if you find it in your lab, you have 7 days to either move it to a lab that can handle this, or you have to destroy it. So we worked with the New York State Health Department, the Wadsworth Center, to do the sequencing because I do not have a select agent lab. And we're able to get isolates from the outbreak and from before, from a bunch of different states, and from several different mosquito species, some horses, and a random turkey. So here's what the phylogenetic tree looks like. Again, this is a time-resolved phylogeographic tree where the colors represent locations. And the 2019 viruses from this outbreak are here in these larger circles. One thing to notice is that, right, when we think about SARS-CoV-2, we think about variants and new genetically distinct viruses that come and sweep and have these big impacts. This was caused by a whole bunch of different lineages, suggesting that this outbreak was not being driven by some new virus itself, right? This outbreak is being driven by what is happening in the ecology, right? When we look at these branches and we start to then take each one of these points here where we see it moves from one location to another location, and we just plot all of those at once on this tree, you see this really interesting pattern here where it all starts in Florida and moves its way north. There is the genetic diversity of this tree is maintained in Florida, and it's constantly being reintroduced. And this is a virus that infects birds. So this is being moved to the Northeast via migratory birds. And this happens quite often. So another way to look at this is, I'm just going to change the color of the tree here just to focus on, here's the clades that are in the Northeast and any place else. And if we look at each one of these clades and just take them and plot them over time, where the first dot here represents when we estimate that that was introduced into the Northeast, when we first sampled it and when we last sampled it, what we see is in the Northeast, these lineages only exist for a couple of years. They cause an outbreak and they go away. The virus cannot be maintained in the Northeast. The virus can be maintained in Florida, but not here. So everything that happens further north is really impacted by this constant reintroduction that is happening through migratory birds. We spent a lot of time, if you want to learn more about this, you can read the paper. There's a lot of time of trying to figure out what might have caused the outbreak. And sometimes in epidemiology, it's really hard to show that something was the cause, but you can rule a bunch of things out. So we ruled a bunch of things out. It's not due to expanded range. Where we're finding EEE in the Northeast is where we found it historically, again, in these swampy areas. The case demographics didn't change for this. Human behavior didn't really change. This is a hard thing to gauge, but we had these cell phone data that helps to get at how much time people spend outdoors. And that has been pretty consistent throughout the study period. Bird abundance didn't necessarily change. There's not like some correlation between where we find the virus and the types of birds that are there, but absolutely bird immunity is probably plays a role here, but that's something that we don't have a whole lot of data on. But what we do have a lot of data on are mosquitoes, because New York, Massachusetts, and Connecticut, as well as Pennsylvania, I know the folks here that do this have great vector surveillance programs where they're constantly going out and collecting mosquitoes and testing them. And so there's some things you can see here. This dashed line is the number of mosquitoes tested. And each one of these programs has set traps. So the numbers of tested means that there's more that were actually collected. And then the bar graphs show the infection rates of the of EEE in these Culicida melanura mosquitoes. New York, interestingly, I'm just going to go back for a slide for a second. There's two main, so here's Massachusetts and Connecticut. This is one foci where there's a lot of transmission, and then you have this other one up here in New York. Here in New York, in 2019, actually didn't seem to be all that abnormal. It was mainly driven by a lot of the things that are happening here. So when you look at the mosquito infection rates, the number of mosquitoes detected from New York, it really wasn't that different. But both Massachusetts and Connecticut, 2019 was exceptionally high, right? So a lot of infected mosquitoes, don't really know why yet. There's a couple of other ways that we can look at this. This is the number of positive mosquito pools that we found for EEE, correlated with the number of human and horse cases, and there's a positive correlation between them. The stars are 2019. These were the high years. Both looking at Massachusetts and Connecticut, where we did see this big outbreak in 2019. It was a very high year for abundance of these mosquitoes. It was a very high year for the infection rate. This vector index is just the multiplication of the two things to think about the risk of the number of infected mosquitoes that are out there. This was very high. And this index P is where we try to take weather data, mainly temperature and humidity, and try to model to see, okay, were the conditions just so right for mosquitoes that this is why? And it's kind of a normal year. So we can't really figure out why yet EEE was so high at this time, which, you know, kind of had us scratching our head here. Like, what we wanted to do is learn so much about this that we can create this amazing model. We can predict outbreaks, give this to public health agencies, and now you have your tool. But can we actually do this? Right now, no, not so much. And one main part is the stochastic aspect of these introductions that come in from Florida. So we need to know a lot more about the dynamics in Florida and how viruses are being introduced and how that timing works with mosquito populations to understand that we can predict, at least far in advance, outbreaks. So this part seems to be a little bit difficult to do, but we're interested in this. But can we use early season mosquito surveillance data to get at late season risk? Human cases happen in the late summer and fall, but infection rates start in the early summer. So this is what we wanted to try to do. If you had some of these early season data from these surveillance programs, can we say is there going to be a lot of EEE this year? So we created this model where we, these are the covariates that went into it, this vector index that I was talking about, the first month of detection, this modeled value, the year, and the month where the cases occur. And this just shows that in October, being in October has a higher risk for cases than being in August, because this is when they happen. When we take all these covariates, we can create this model where the actual values here are in black and our predicted models are here in red. The model works pretty well. And the main thing that we got from this is that if you detect any positive mosquitoes in July or in June, there's a 220% increase in the likelihood that there's going to be cases in October. That has been put to the test this year because now we're in 2023. This is 4 years ago. EEE goes in these weird cycles. This is a really big year for infected mosquitoes with EEE in Connecticut. This was a weird year. There was a lot of rain in July. I think we broke the record for rain in July. It's been a hot fall relatively and still kind of wet. We're still detecting positive mosquitoes, but we didn't detect any of them until August. And we have yet to have a human case. We had an emu that was detected. I don't even know how that someone tested an emu, but there was an emu that had it, but not a human yet. So hopefully that these were late season, that eventually the mosquitoes will start to decrease as we get into fall, and there's not enough time to have these severe cases. So we'll see if the model actually works in our predictions here. So how do these outbreaks occur in the Northeast? Well, they're self-limiting. You have an introduction of a virus that comes in from the South. It starts local transmission chains. It finally builds up when you have susceptible birds and a large enough mosquito population, and boom, you have the right conditions for an outbreak. The potential for outbreaks always depends on these new introductions. And probably with waning, with bird immunity that wanes, then you have now the population susceptible for this to happen again. And currently, this is not predictable outside of a couple of months, but we're hoping to get data from the Florida Department of Health to combine into these models. What do you think for another vignette? Do we have? Okay. So the last one here, this is, we're going to talk a little bit less about ecology and more on the human aspects of things. My group does a lot of work studying dengue virus and building tools to create a global dengue virus genomic surveillance system. I work with the Florida Department of Health on this. Y'all might have heard that in Pasadena, they had their first ever locally acquired dengue case this past week. Last year in Arizona, they had their first ever locally acquired dengue cases. In Florida, there are locally acquired dengue cases, and of course in Puerto Rico and Hawaii. This is becoming more and more of an issue in the United States. This is a project that's led by a pediatric fellow, infectious disease fellow that works in my group, and a whole bunch of collaborators from the Florida Department of Health and the CDC. So dengue, the reservoir of dengue are in these sylvatic cycles where it's transmitted by these forest-dwelling mosquitoes and non-human primates. About 100 years ago or so, 4 different events, at least that we know of, where this sylvatic virus has spilled over into a human population and has evolved the 4 distinct serotypes of dengue virus that now exist throughout the world today, and they're transmitted, and they're by Aedes aegypti, which is a urban mosquito. It basically lives in environments where human lives. They almost exclusively feed on humans. They like to feed on rubbish and all sorts of other things of any sort of, or sorry, they breed in any sort of standing water that might be left by plants or trash or anything else that is around. And so the virus now has this great cycle where you have this, the host that is readily available in these dense urban environments and lots of mosquito populations. It exists throughout the tropics. And as I mentioned, now it's encroaching more into the United States. One of the regions that I'm really interested in is the Caribbean. One is it's really great environment for dengue to exist. And because of the tourism in the Caribbean, it's an important place for the dissemination of the virus elsewhere. And in fact, in Florida, anytime there's an outbreak in Florida, it is coming from the Caribbean. This outbreak, these a couple cases that happened in Arizona, these were also coming from the Caribbean. But we know very little about what is actually happening with dengue in the Caribbean because a lot of the low resource settings of a lot of these Caribbean islands, they just don't have the infrastructure to do a lot of dengue surveillance, so we can't get samples to use for sequencing and other sorts of things to understand the dynamics. So working with the Florida Department of Health, they get travelers. So basically humans as sentinels, right? You go out into these sites and you experience all the local culture, you get bit by mosquitoes, you come back to Florida and you happen to bring with you a souvenir that you probably didn't intend to have. You get diagnosed in Florida, and we can then use that information to understand more about where you actually got infected. And so can we use travelers to reconstruct dengue virus in the Caribbean? And can we use this to detect anything, major events that are happening, like new introductions that would be into the region that could be of importance for all of the Americas? So from 2009 to 2022, we gathered data and samples from the Florida Department of Health. They get a lot of traveler cases, as you can see by this map here, that most of them come from Cuba. A lot of people visiting family is the primary source of this. And then several other, the top 5 are all Caribbean islands of where the travelers are coming from. So here, we looked at these cases every single year, and we just wanted to know, are the travel associated cases for where these people are getting infected versus their local cases, do these match? This is what we're looking at here. So these, this grayish brown is the number of local cases that have been reported by each place per year. And then the darker lines are the number of travel associated cases. And then when we normalize that by either the total population in the country for the local cases or by the number of travelers, we get a lot of airline data from the CDC's Department of Global Migration, and we can use this as the denominator here. And we see that for places like the Dominican Republic, Jamaica, and Puerto Rico, this correlation between the local and the travel cases is almost perfect, right? And we know from these locations too, they have really great dengue surveillance programs. We did a study with Zika using travelers a while back, and these locations as well had very great correlations between local and travel. We have 2 places where we don't have great correlations. One of them being Haiti, and Haiti does not have a great surveillance system for dengue, in part because of the low resources there, all the natural disasters that happen there. There's just a lot of things that prevent these sorts of systems from being built up. And so they're not reporting cases, but it doesn't mean they're not having cases. The other one that's always been interesting in this work has been Cuba. Cuba has a fantastic surveillance system, but they're not always so interested in sharing those data with the international community. And sometimes, honestly, what I feel like that they are sharing on these sites, like the Pan American Health Organization, just a random number generator. And so they don't always really line up very well with our travel data, and it doesn't always line up very well with a lot of other data, not just for dengue. So can we use this information where we have good data to help us to understand where we don't have great data? So again, we created a model where we have the observed local cases, and what we predict would be the local cases based on the surveillance data, also using some other covariates like the gross domestic product. And so we trained our model using these places, and then we applied it to Cuba and Haiti. And one of the things that stands out here from this is that in 2022 in Cuba, there was this very large underreported outbreak that we are detecting that is would have been the largest that we've detected in Cuba for at least the last 10 years, and one of the largest in the Caribbean. Now, it's not so large compared to some other locations where they have more intense dengue transmission, so it's not a complete outlier here, but it was quite concerning. So we wanted to know, I'm not going to go into the entire story for the sake of time, we just wanted to know a little bit more about what is happening here. So we sequenced 295 travel cases, and there's 4 dengue serotypes, so these have to go into 4 different trees. The red cases are Cuba, and right here in dengue 3 is where we're seeing a whole bunch of these 2022 cases. So if we zoom in on this part of the dengue 3 tree, there's a couple of things here. So Cuba is here in red. These other ones that we've sequenced from travelers is here in green in Jamaica. These viruses here that are shown in this part of the tree typically existed in Southeast Asia. And what we have detected here, both happening once and twice in 2014 and 2020, are 2 separate introductions from Asia into the Americas. Some of you may be thinking, yeah, of course, people travel all over the place. There's going to be introductions. But what's actually interesting is that they're not bouncing back and forth between the Americas and Asia all that often, right? It's actually kind of a rare-ish event that might actually be increasing more in frequency. But this is not happening. We're not seeing this every year. It's not like SARS-CoV-2 when something pops up in one place and is everywhere within months. This is kind of a rare thing to happen. So this first time that this lineage was introduced into Jamaica, we estimate that it persisted for a few years, caused an outbreak in 2019, spread here to St. Lucia, and then we haven't really detected it since, so we're not quite sure what's going on. This other one that came into Cuba that we think is causing the majority of this very large outbreak was introduced in 2020. This time of 2 years from outbreak or from introduction to outbreak for dengue is really fast. Usually it takes, I don't know, 5 or 6 years for something to get introduced and slowly start to build up, and then it causes an outbreak. This suggests to us that the population was very susceptible to this. This is the same, it's hard to, we focus on the travelers here. Within this clade is actually this was a bad outbreak year for dengue in 2022 in Florida. They were all in this clade. Those viruses that I was saying, the local transmission in Arizona, were these, we have now detected this virus, the same one here in Brazil and in Peru. And in 2023, dengue 3, which is this, cases are going up. And so we're concerned here that this has an advantage in the Americas, mainly because it might be divergent enough from what people might have antibodies to. And so they may not be protected against this version of dengue 3 or some other things that I'd be happy to chat about later. So this just shows the importance of being able to use travelers to detect things that we wouldn't have known. We reconstructed this entire outbreak from Cuba only out of travelers. There's no other information here that would have suggested that this happened. So to end this last vignette, travel surveillance that I really, really enjoy doing, just being able to concentrate some of your efforts and get a snapshot of what is happening around the world. This is great to uncover some of our blind spots in our surveillance systems. It can provide this global snapshot of genetic diversity. And as dengue vaccines are about to be rolled out here, this information is going to be very useful. And it can help to detect these important epidemiological events, like I was just suggesting. But it absolutely cannot replace local surveillance. Right? This is a supplemental tool, not something that can replace anything that is happening on the ground. So I hope I convinced you all the power of genomics to help uncover patterns of emergence and spread. It has the potential to point for where interventions can be placed to help decrease the risk to humans, and why we need this as a very routine part of our public health system. And with that, I have a whole bunch of different collaborators that I'm not going to list all their names to recognize here, but specifically Verity Hill for the Tripoli work, Emma Taylor-Salmon for the dengue work, and Chantal Vogels for the Poisson work. The dengue work, at least, was supported by the NIH. The Tripoli and Poisson work, we just been doing on our own dime because we think it's interesting, and it's been more fun than studying COVID right now. So it's been a nice aside. If you are interested in all of our data, we're completely open with sharing and all of our tools. I'll show you our website on the next slide, but if you go to our website and go to this tab that says Next Strain, you can find all of our data and play around with it. Here is my website and my information, though I'm not really on Twitter that much anymore. But if you do have other questions, feel free to email me, and thank you all. Yeah.
Very interesting. I have a question. So first, in your dengue story, since those are endemic regions, and this was the antibody enhancement, is the introduction from Asian strain, are they just getting better as a subsequent infections, or are these naive infections?
So throughout the Americas, we don't really know. It's probably a little bit of both. For some of y'all, so dengue is weird where if let's say you get dengue 1, right, and you're exposed again to dengue 1, you would be protected. But if you get exposed to dengue 3, it might actually cause a worse dengue 3 disease. And this is why it's actually really hard for a vaccine. In this case, it's hard to tell. I do not have any evidence that this is what is called antibody dependent enhancement that is making the disease worse, and therefore we are detecting it more. That could be happening, or it just could be causing more general acute infections. I don't really know the answer to that yet.
And the second question is, from your point of view, how do you feel about vector control, especially mosquitoes, right? So certainly there could be unintended consequences. So like in malaria, they try to introduce engineering mosquitoes that maybe devastate.
Yeah. So without a vaccine, vector control is our only method of really, to reduce dengue, besides like general information about how to avoid mosquito bites. But when you live in some of these places, and it's almost impossible to do. So vector control can provide some immediate relief, but it is not a very good long-term solution. There's lots of new methods now of releasing genetically engineered mosquitoes, or my favorite are these mosquitoes that are infected with a bacteria called Wolbachia that make them not very good at transmitting the virus. These all show very good, early clinical results that they can reduce pediatric dengue cases. But it's really hard to know what is the long-term impacts. And you think about, I have no idea how many mosquitoes there are in this world. It might be approaching the number of stars. I don't know. But it would be very difficult to replace all those populations and the other unintended consequences. We need a vaccine. I think that's just incredibly clear.
So I was just curious about your triple E story. I'm just wondering, is the mosquito line, the vector line for triple E, essentially going just south of the northern part of Massachusetts and Connecticut? Because I can't explain when you list some migrating birds go much more north.
Mm-hmm.
So it would really be why not more north if you have triple E? So it's only on the voyage and we stop somewhere at the place where now we begin to have the vector.
Yeah.
We begin to get the effect.
Yeah. So this gets into like the, I don't know, the Goldilocks effect here of like, you have to have all the perfect conditions for this to happen. I think migratory birds will make several stops and then will get fed on by the local mosquitoes along the way. And then it has the opportunities to, you know, offload the virus into the mosquito populations. Triple E does go up into Canada as well, but then you start to get into some conditions that don't make it great for those mosquitoes to exist. And they found this little happy area in the Northeast as well of like, you know, swampy, warmish areas where they can kind of thrive. But Triple E is found throughout the eastern part of the United States. And in fact, there are cases in Michigan and other places in the Midwest as well. I think it's another one of these things is expanding, but not very many surveillance systems actually look for it. So we don't really know the total range here, but basically wherever you can find Culicida melanura, you can probably find this virus in that mosquito. All mosquitoes and ticks and stuff are constantly kind of changing their you know, home range.
So the two plagues on Powassan, relating to Powassan,
Mhm.
you said one of them is in the Midwest.
Mhm.
The other one is in the Northeast. Can't help but notice that Ontario's right in the middle. Do you think they're both from that original index case?
Well, not derived from, because that the case in Ontario in Powassan, Ontario, where this is named after, was that the 50s or 60s. That was actually a lineage 1 virus. So that would have been a whole separate clade. So the ancestors to all of them for both of those viruses were probably like 10,000 years ago. And they're related to other tick-borne viruses from Russia. So this might be talking about like Lambridge sort of stuff of when they actually came over. They're quite divergent, but they're serologically related, so they're all one virus, but not so much related to what was happening in Ontario. But that the lineage 1, this more of this cryptic forest virus, we actually thought was extinct because it hadn't been found in like 40 years. But it turns out nobody was looking for it. When we started testing some ticks for it, we found it. It just doesn't really cause very many human cases. So it's just kind of chance that was the first one detected. And of course, it's one that doesn't cause very many human cases. Yeah.
This Powassan virus is actually, you know exactly where the Lyme disease that we get the most early on. I'm just wondering, did ever anybody check for the ticks? Easier to infect the virus when they already have the Lyme.
So there's always one thing guaranteed about my talks about Powassan is something about co-infection with ticks, because this is fascinating. If a bunch of different pathogen systems are being maintained by the same ticks, what does co-infection do here? And the answer is we don't really know yet. I mean, there's not very many experimental studies that are happening to actually look at this. The Powassan foci, and because they're so centered around smaller areas versus like Borrelia for Lyme disease is a little bit more ubiquitous in ticks. We don't find a whole lot of co-infections in ticks, but maybe people just aren't looking at that. So your guess would be as good as mine if co-infection actually plays a role there.
Because at Penn, we have Rusty Risk, who's working in Lyme disease, so he will guide to us.
Yeah.
When it sticks, which he tests every year.
Yeah.
Not thinking.
Yeah. So most people who study Lyme disease and they test ticks for Borrelia don't pay attention at all to Powassan. And then the other way around, people who focus on viruses don't look at the bacteria as often. We're writing a grant right now where we're trying to bring in people from both sides of things to think about this holistically. Because I think you're right. We need to have a better understanding of how. There's one part of the US where you have Lyme disease, you don't have Powassan, and that's in the Southeast. And that's because Lyme is maintained in a different sort of system and doesn't really use as much of the small mammals, so Powassan can't exist there. I have this weird thing with lizards. Oh, pull that up. How many Triple E cases are there in Florida? I don't know as much on the, I haven't looked at the data to tell you how many human cases of Triple E there are in Florida. Oh, that is the wrong direction here. But it's less cyclical than what is happening in the Northeast, where we have these periods where we don't find it and then boom, a whole lot. It's a little bit, it's more stable that you'll find it. It's not more than a handful of cases a year that you end up finding. And again, because I think it's just the tip of the iceberg, the ones that you see are the really severe cases.
If there are no more questions, let's thank Nathan for a wonderful talk. As a small token of appreciation, the executive committee and the branch is presenting him with a copy of James' book, and that's generously by Barbara. So
Thank you so much. This is really awesome. Thank you.
And so second thing is that we have those parking validations, and Jason has them. And so if you need a parking pass, go to him. I think free for students, $5 for members. And we hope to see you at PIF or the clinical symposium, both in December. If not at the very least, I will see you January, the next monthly meeting. Thank you very much for coming, and have a safe drive home.